Hello everyone. In this video lecture, we continue covering somatic sensory division, specifically special senses, and we will focus today on the physiology of sensation. So I already covered the beginning of this PowerPoint. So if you're interested in anatomy of the eye or the ear, please watch my previous lecture. But today we're talking about physiology. So let me pick up my pen. Okay. And let's go ahead and begin. It's interesting. I always pick up my pen and then I realize that I need to change my slide. So I need to go back to the mouse and then pick up the pen again. Sorry for that. Okay. So a little bit about physiology of gustation, that is your taste. So on this picture over here, you can see your tongue, that is a special organ for gustation. So it's a special sense. And you have a taste bud. And if you look inside this taste bud, you can see several cells. Well, some of them are supporting cells, like this one shown here in yellow. But other cells that are here in different colors, they are receptor cells. And receptor cells, they respond to a specific chemical. So that chemical will activate receptor cells. They will create a receptor potential. A receptor potential is really electrical impulse inside the cell. So when we create receptor potential, that will cause a release of either neurotransmitter, for example, over here, serotonin, or release of ATP. And those molecules, ATP and serotonin, they're going to activate the neurons. And these neurons will deliver information about taste to your cerebral cortex, right? So those receptors shown over here, this receptor specifically is for sweet taste. And you can see that embedded in a cell membrane that there is a receptor for sugar molecule. When sugar molecule binds to this receptor, it activates a protein inside the cell that is called G protein. This activation will cause chain of chemical reaction, and those reactions will activate another molecule called IP3, that is second messenger. Now, when IP3 is activated, it binds to its own receptor on endoplasmic reticulum, and this causes a release of calcium. Now, when calcium is released from endoplasmic reticulum, calcium binds to sodium channels, and it causes sodium channel to open, and sodium start moving in. When sodium moves in, it depolarizes the cell. So remember, at rest, cells of your body, they are negative inside and positive outside. And this separation of charges is called potential. It's like in your battery. When you separate positive and negative charges in your battery, you create some potential energy, right? The same is true for your cells. Your cells separate negative and positive charges across a cell membrane, creating what we call a resting potential. But when positively charged ions move inside, then guess what? Cell becomes positive inside, and this is called depolarization. Because cell is polarized, it's polar, it's negative inside, positive outside. Depolarization means you reduce polarization. So on a graph, you move towards zero voltage, right? So that's depolarization. And depolarization of receptor cells will create receptor potential. And receptor potential, again, in this particular example, will cause ATP to be released and ATP activate your neurons. Now, when ATP activate your neurons, inside your neuron, you create now action potential. And action potential is an electrical impulse that will travel through dendrites, cell body of neurons, axons, and eventually reaches your brain. And you can feel the sensation of sweet or salty or bitter. Right, so that's how we have processing of the information from sugar molecule, right? So here's our sugar molecule to your brain, right? So the sugar molecule does not travel to your brain, right? The sugar molecule, what it does, it activates your receptor cell. So receptor cell create electrical impulse, releases neurotransmitter or ATP. Neurotransmitter and ATP create action potential in your neurons. That is another electrical impulse. When action potential reaches your cerebral cortex, it activates your neurons of your brain, and you process this information as a taste, right? Okay, so that's quickly about gustation. Next, we will talk about your ability to hear or audition. You know, you have a special organ for hearing, and this is your ear. So your ear is divided into external, middle, and internal ear, and all three parts are involved in hearing. So if you look at this diagram, you can see that the sound waves travel through your external acoustic meatus or auditory canal, and sound wave is mechanical wave. That means it has ability to vibrate stuff. So it's gonna vibrate your tympanic membrane right there. Tympanic membrane is attached to auditory ossicles. Those are little bones inside middle ear, malleus, incus, and stapes. And vibration of tympanic membrane will vibrate the auditory ossicles, and purpose to do it is to amplify the sound, to make it stronger, the wave. Then pressure wave created by those ossicles and stapes is a part of the ossicle, will be, so that a pressure wave will push on the oval window. Remember, we have oval window over here, and oval window will transmit this vibration through the scala vestibuli. Now, this is cochlear over here. Inside the cochlear, you have three channels: vestibular channel, cochlear duct, and tympanic channel, or you call it scala. So you have scala vestibuli, you have scala tympani, that is this part below, and of course in the middle you have cochlear duct or scala media. So now pressure waves created by stapes pushing on the oval window move through the fluid in a scala vestibuli. So here now you have movement of the fluid, and this is perilymph inside scala vestibuli. Now sounds with frequencies below hearing travel through the helicotrema. So some sounds, they kind of moving through the top part of the cochlea. This is called helicotrema. And they do not vibrate the membrane inside the cochlear duct. So for you to hear sound, you need to vibrate what is called over here basilar or basilar membrane inside this cochlear duct. So here's your cochlear duct, and this is your membrane over here that is basilar membrane or basilar membrane. So if you want to hear a sound, this membrane need to be vibrating. So like it's shown over here. So sounds that below our hearing range, they do not vibrate basilar membrane. So they go through the helicotrema, and then you don't want these waves be stuck inside the closed surface. That's why they kind of moved out back to middle ear through round window. But the sound in the hearing range goes through the cochlear duct, so you can see they go right here, and vibrate basilar membrane, deflecting hair cells, and hair cells located on this basilar membrane, and that allow you to hear sound, right? So again, very quickly, sound waves vibrate tympanic membrane vibrate malleus, incus, and stapes. Stapes vibrate perilymph inside the scala vestibuli. This vibration, if it goes through the cochlear duct, it's gonna vibrate basilar membrane. When basilar membrane vibrates, the hair cells located on this basilar membrane, they get excited, and hair cells are our receptors. When receptors for hearing are excited, they will pass this excitation or this electrical stimulus impulse to your cochlear nerve, and cochlear nerve will deliver this action potential or electrical impulse to your brain, and you will be able to hear stuff, right? So now let's see how we can determine the frequency. So high frequency sound displaces basilar membrane near the base, right? So you can see these waves are very high frequencies. Those are medium frequencies. So they displace basilar membrane near the midline, and low frequency sounds displace the basilar membrane near the apex. And depends where your basilar membrane, that is this one on the bottom, is displaced, where your hair cells are activated, your brain interprets it as a high frequency, medium frequency, or low frequency sound. Okay, well, now we're looking into more details. So we are inside this cochlear duct. So here's our basilar membrane, right? So right here, that will vibrate, right? So when this basilar membrane vibrate, then we can hear stuff. Now, what is located in this area is called organ of Corti. So that's our organ of Corti. So it's located in cochlear duct. And organ of Corti is made of supporting cells, outer hair cells, right here, and inner hair cells. Also, those hair cells, here they have what we call stereocilia. Those are not really hair. Those are cilia or microvilli type processes of a cell membrane. So covering those hair cells is another membrane that is called the dictorial membrane. So give me just 1 second over here. I want to, yeah, sorry for that. Okay. Now, when basilar membrane vibrates, right? And it vibrates at a specific spot, right? So it can be vibrating close to the base of the cochlea, middle of the cochlear duct, or the apex of the cochlear duct. So when it vibrates based on the frequency of the wave, then those hair cells vibrate too. So you can imagine that when this goes up and down, now this hair cells also go up and down. And because this stereocilia is attached to tectorial membrane, this cilia will bend, right? So you bend it either one way or you bend it another way. And when this bending happens, those receptors, or hair cells are receptors, they are excited. And when they excited, they will release the neurotransmitters that will activate a nerve, cochlear nerve, and cochlear nerve will send this action potential or electrical signal to your brain, right? So that's how things happen. Now over here, we can even see more details of this process. Now those, so what do you see over here? So you see individual hair cells. So here's one hair cell, here's another one, right? Now, those processes over here are not hair. Their name is stereocilia. So here's the stereocilia. Then one is stereocilium. And they're not hair, they're not even cilia. They are microvilli-like structure. They extend to tectorial membrane. So tectorial membrane gonna be right here on the top of those stereocilia. Pressure waves on the basilar membrane will cause those cells to move up and down, and then tectorial membrane slides across stereocilia. So you can see the tectorial membrane that will slide across those stereocilia causes stereocilia to bend, right? And when stereocilia bend either one direction or another direction, some changes happen within our receptor cells. Now, well, first of all, let's look what's going on at rest. So this is your hair cell at rest, and about 10% of the ion channels are open. And ions, specifically in this situation, potassium ions. So potassium ions slowly are moving inside this hair cell and create this tonic signal. That means that at rest, your hair cells release neurotransmitter. It's and when neurotransmitter is released, look, you activate your cochlear nerve, but it's a tonic signal. So at rest, tonic signal is sent by the sensory neurons, right? So here's your sensory neurons to your brain. And this is how it will look on the graph. Now, tonic signal is an action potential. So here's action potential that sent through the sensory neuron, and your brain is your brain does not interpret it as a sound, right? For your brain, this is silent time, right? So it's tonic signal is processed as nothing. Now what's gonna happen when this basilar membrane vibrates and tectorial membrane slides across the stereocilia? Then the hair cells bend. Well, when they bend one direction, more ion channels open, more potassium moves in, and that causes depolarization. Depolarization means the cells became positive inside, so more neurotransmitter is released, and action potential increases in frequency, right? So, we bend towards one direction, right? That causes depolarization because more ion channels open, more potassium in, more neurotransmitter is released, and frequency of action potential increases. So that's excitation of hair cells. We also have inhibition of hair cells. Inhibition, if the hair cells bend in the opposite direction, then the ion channel closed. We don't have influx of potassium, so no influx of potassium, no release of neurotransmitter, and the neuron is inhibited, and this will cause, so it will cause polarization and decrease in the frequency of action potential. But the point is, when we have increase or decrease in the frequency of action potential, this is change compared to the tonic release, and this is what your brain process as sound. Okay, this is how, it works. Let me see what is the next slide is. Now, equilibrium. All right, so we pretty much, let me just exit to show you that we talked about transmission of sound from external to internal ear, right? We also said that when sound waves, when they move through the perilymph, they vibrate the basilar membrane, and based on the frequency of sound, basilar membrane is vibrated at a different points, and this is how you can hear high pitch and low pitch, right? A high pitch like this, or low pitch like this. You see, when I talk to you like that, your basilar membrane is vibrating at a different points, close to the base, in the middle, or to the apex, right? We also said on this basilar membrane, we have this organ of Orti, that is a sensory part that have outer hair cells, inner hair cells. Actually, only in inner hair cells are involved in a sounds transduction, and outer hair cells have other important functions. But when basilar membrane vibrates, and this tectorial membrane slides and bend hair of your hair cells. That are actually not hair. We call it stereocilia, but they are microvilli. And when those microvilli or stereocilia, that's the name, when they bend one direction or another direction, this will either open or close potassium channels, and either more potassium in, more neurotransmitter released, action potential frequency increases, or less potassium in, less neurotransmitter released, and frequency of action potential decreases, right? And your brain has this ability to interpret those frequency of action potential and process that as a sound. Okay, now we talk about equilibrium. So let's see what equilibrium is about. Now, for equilibrium, you only need your inner ear. So in your inner ear, you have this structure shown right here. And it's made of cochlear, right, this part, that is for hearing. So now we're not going to talk about cochlea, because we're not talking about hearing. And two other parts of the structure are vestibule and semicircular canals. So please watch my previous video when I cover in detail structure of vestibule and semicircular canals. Both of them are for equilibrium. Now in the vestibule, you have structure called utricle and saccule, and inside this utricle and saccule, you have macula. And macula is a sensory organ for equilibrium. inside semicircular canals in the ampulla, you have crista ampullaris, and crista ampullaris is for equilibrium as well. The difference is in utricle and saccule, this macula is for static equilibrium. So it's shown over here. Just when you, for example, bend your head up and down, when you move front and back, left, right. And crista ampullaris inside the semicircular canals is for dynamic equilibrium, or for example, when you rotate your body. Now let's look at the macula. So here you see that's a macula over here, and our hair cells again look familiar, right? So here's our stereocilia. Now on the top of this stereocilia, you have very thick fluid, and inside this fluid, you have tiny stones made of calcium carbonate. So here you have these little stones that their name is otoliths. Right. Now look what happens. So here's your head is upright, and the whole structure is called otolithic membrane. So this otolithic membrane is pretty much upright as well, and hair cells are not bended. You see, they're looking up straight. Now, when you bend or you tilt your head, so here you tilt your head forward, and look what happened. Because of the gravity, this otolithic membrane are kind of sliding down. It's heavy. It has all these little stones, so it slides down and it bends the hair. Yeah, that's what we need. When we bend the stereocilia, we open or close ion channels. So here, let's look on the bottom part, right? So here's our hair cells that are our receptors with stereocilia. Now, I forgot to tell you that the biggest, kind of like biggest stereocilia are called kinocilium, and stereocilia bends towards kinocilium or away from kinocilium. And receptor cells, synapses with neurons of vestibular nerve. Now look what happened when your head is upright. So when your head are upright, we have this tonic innervation, tonic activation, I would say, right? Tonic activation of the neurons, similar to hearing. And we do send some action potential to your brain in this tonic pattern. Now, when hair bends towards the kinocilium, the hair cell depolarize. So that means potassium moves in, and it excites your, when a lot of potassium moves in, it causes receptor potential. So hair cells release neurotransmitter, and it's activate your neurons. So it excites the nerve fiber, we generate more frequent action potential. So we increase the frequency of action potential. Now, when your hair moves in the opposite way, ion channel close, we have hyperpolarization. I always want to say some different word. Hard word for me with my accent to pronounce. Hyperpolarization. When hair cells bend, when stereocilia of the hair cells bend away from kinocilium, the hair cell hyperpolarizes, inhibiting. So then, no potassium in, no neurotransmitter. So it's inhibition of nerve fiber that decrease the frequency of action potential. And guess what? When it's tonic stimulation, your brain interprets it as head is upright. When we have increase in frequency, your brain interprets it as, okay, head tilted forward. When we have decrease in the frequency of action potential, your brain interprets it, okay, head tilted backward, right? So that's how this change in the frequency of action potential give your brain information about your head position, and and that's the job of macula, and macula is located in utricle and saccule, and utricle and saccules are located inside vestibule. Okay, all right, next. We also have another organ for equilibrium that are semicircular canals, and they are for dynamic equilibrium, or what we call rotatory movement. So when you actually rotate your head. Now, the principle is the same. So in this semicircular canal, this wider part that is dilated part. So this wider dilated part over here called ampulla. So here you have ampulla. Inside the ampulla, of course, you have fluid. And here's our crista ampullaris. So that's our crista ampullaris right there. This is a sensory structure. And inside this crista ampullaris, we have, guess what? Hair cells again. Surrounding those hairs, that is stereocilia, is cupula. Now, cupula is also like very, very thick fluid. This time we don't have this otholates, but we just have this jello consistency cup on cupula. So when you rotate your head, because you have the fluid, right, and the lymph over here, the fluid moves. When fluid moves, it's gonna move the cupula. And when cupula moves, it will bend hair cells. And here the process is similar to to what we already discussed. When stereocilia or hair bend, then it causes either opening or closing of ion channels, and that will cause either more or less ions moving in, either increase or decrease of neurotransmitter release, or either increase in the frequency of action potential or decrease in the frequency of action potential. And that's the signal to your brain that there is some movement. Okay, and the only thing here I want to mention that cupula deflected in direction opposite to head movement. Okay, that's pretty much it. And of course, also I want to mention again that utricle and saccule are for static equilibrium, right? When there is no rotation, you just bend or tilt your head. And crista ampullaris inside semicircular canal is for dynamic equilibrium or for a rotatory movement. Okay, so I think I'm gonna stop this video here because it's already pretty long. We covered few slides, but I hope we covered in good details. And I will record one more video where I cover physiology of vision and just the central processing, how you process it in your brain, right? So thank you for watching this video. I hope it was helpful. And you know what? If you watching till the very end of this video, then please click like. It means you like this video, right? Or if you don't like it, don't click like button. Okay, I talk to you again in my future videos. Have a nice day.